Good afternoon, dear colleagues. I'm glad to see everyone, and let me share the presentation with you. Can you see the slide properly?
Yeah.
Yes,
Okay,
ma'am. Your slides are visible and you can start with your presentation.
Okay, thank you. My name is Maria Dobrinskaya. I'm from the Ural State Medical University. Let me present my topic: toxicological profile and regeneration activity of new doped nanocrystalline carbonate-phosphates for bone grafting. So nowadays, the problems of modern medicine and biotechnology involve not only creation of implants replacing bone tissues and organs, but also synthesis of biologically active materials promoting the fullest restoration of tissues and maintenance of necessary functions of the organism. So, we are focused on the materials of doped calcium phosphates. Calcium takes place in the numerous metabolic processes in our body, and our bone tissue contains over 70% of calcium hydroxide phosphate or calcium hydroxyapatite, and 30% of the organic component like collagen fiber. And also the ions as potassium, sodium, magnesium, ferrum, and chloride are contained in the structure of calcium hydroxide phosphate of the bone tissue. Besides calcium ions and phosphates. Therefore, in the view of carbonate groups introduced in the structure of calcium hydroxide phosphate, its possible formula will be as follows. And the vacancies can be filled with bivalent cations of trace elements received by a living organism with anions like CO2, CO4, or chlorine or chloride ions. So we study the materials, new ones, nanocrystalline calcium carbonate phosphates, doped with iron, magnesium, and silicon ions. The picture, how it looks like, is here in the slide. The formation of hydroxycarbonate complexes from three calcium carbonate polymorphs like calcite, tartaride, and aragonite. In the solution of ammonium chloride and ammonium carbonate, followed by reaction of orthophosphoric acid. And then we provided experimental work using these materials to prove whether it's safe for the organism. And experimental work was conducted using healthy male and female white mice, Vistro rats, chinchilla rabbits, which were carried out with the standard diet and standard conditions. So we investigate the acute toxicity in white mice. We injected new material as the 10% suspension and 30% suspension of new doped carbonate phosphate intragastric and intraperitoneally. And also we had the control groups with the sodium chloride injected or gavaged. Then we used the experimental work on the Vista rats. Also, we provided to conduct acute toxicity in Vista rats. They were also gavaged, 50% suspension of doped nanocrystalline calcium carbonate phosphate. And they were given it intraperitoneally as a 10% suspension. And also we had control groups, which were given 0.9 sodium chloride solution. Also, we defined whether our new materials had local irritation effects, sensitizing and skin irritating properties. And we discovered regenerative potential in bone tissue. We made the fracture model in rats. We fracture model of tibia and fibula was set on Vista rats, and they were divided into two groups, experimental one and the control one, without any materials inside the fracture area. So in the experimental group, we put the substances in the fracture area. And also we conducted biocompatibility studies. The test samples were saturated under the skin in West rats. And we also made the histological studies finally. As the result, no animal mortality or abnormal behavior was observed in any of the task group of the entire period of the study. No locally irritating or sensitizing effect on the study substances were detected. And the histological studies show us that in the control group, we had such a picture. The connective tissue formed around the sample of doped nanocrystalline calcium carbonate phosphate with the which was saturated under the skin, and we can see the loose connective tissue with functionally active fibroblasts and sinusoidal capillaries. Number 2 is sinusoidal capillary, and number 1 is just the loose connective tissue. And if we look on the picture of the experimental group, we can see the connective tissue formed around the sample of doped nanocrystalline calcium carbonate phosphates. And we can see the focus of dense fibrous tissue and well-formed vessel inside. These are the pictures of histological samples of the organs of the experimental groups. So we had no changes, abnormal changes in the structure of any organs, liver, cardiac muscle, adrenal, kidney, and spleen. The structure was pretty normal. And the area of the fracture showed us that in the control group, we can observe the focus of cartilaginous tissue. It is not well formed yet. And at the same time, we have the experimental group where the area of fracture has the normal lamellar bone and normal osteon formation. So in conclusion, we can say that toxicology analysis of doped nanocrystalline calcium carbonate phosphates confirmed its safety for laboratory animals, and the result indicated that the nanoparticles of the calcium carbonate phosphate doped with cations of iron, magnesium, and silicon did not exhibit any toxicity and possess a remarkable regenerative potential, which give a prospect for further preclinical and clinical research. Thank you for your attention and welcome to URLs. Thank you. I'm ready to answer any questions you have.
Thank you for completing a valuable presentation at our platform, Ms. Maria Dobrinskaya. If any other participants have any doubts to be cleared regarding this presentation, it is the time for the question and answer session. You can ask now. As there are no questions, we are moving on to the next presentation.